and functions. Biology is the study of living organisms. The detailed description of their form and appearance only brought out their diversity. It is the cell theory that emphasized the unity underlying this diversity of forms. The cellular organization of all life forms. A description of cell structure and cell growth by division is given in the chapters comprising this unit. Cell theory also created a sense of mystery around living phenomena, physiological and behavioral processes. This mystery was requirement of integrity of cellular organization for living phenomena to be demonstrated or observed. In studying and understanding the physiological and behavioral processes, one can take a physical chemical approach and use cell-free systems to investigate. This approach enables us to describe the various processes in molecular terms. The approach is established by analysis of living tissues for elements and compounds. It will tell us what types of organic compounds are present in living organisms. In the next stage, one can ask the questions: what are those compounds doing inside a cell, and in what way they carry out growth, physiological processes like digestion, excretion, memory, defense, recognition, etc. In other words, we answer the question: what is the molecular basis of all physiological processes? It can also explain the abnormal processes that occur during any diseased condition. This physical chemical approach to study the understand living organisms is called reductionist biology. The concepts and techniques of physics and chemistry are applied to understand biology. In chapter 9 of this unit, a brief description of biomolecules is provided. G.N. Ramachandran, an outstanding figure in the field of protein structure, was the founder of Madras School of Conformational Analysis of Biopolymers. His discovery of the triple helical structure of collagen, published in Nature in 1954, and his analysis of the allowed conformations of proteins through the use of Ramachandran plot rank among the most outstanding contributions in structural biology. He was born on October 8, 1922, in a small town not far from Cochin on the southwestern coast of India. His father was a professor of mathematics at a local college and thus had considerable influence in shaping Ramachandran's interest in mathematics. After completing his school years, Ramachandran graduated in 1942 as the top ranking student in BSc Honours Physics course of the University of Madras. He received a PhD from Cambridge University in 1949. While at Cambridge, Ramachandran met Linus Pauling and was deeply influenced by his publications on models of the alpha helix and beta sheet structures that directed his attention to solving the structure of collagen. He passed away at the age of 78 on April 7, 2001. Cell, the unit of life. When you look around you see both living and non-living things. You must have wondered and asked yourself, what is it that makes an organism living? Or what is it that an inanimate thing does not have which a living thing has? The answer to this question presence of the basic unit of life, the cell in all living organisms. All organisms are composed of cells. Some are composed of a single cell and are called unicellular organisms, while others, like us, composed of many cells, are called multicellular organisms. But what is a cell? Unicellular organisms are capable of independent existence and performing the essential functions of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, cell is the fundamental structural and functional unit of life or living organisms. Anthony von Leeuwenhoek first saw the and described a living cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. Cell theory. In 1838, Matthias Schleiden, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissues of the plant. At about the same time, Schwann, 1839, a German zoologist, studied different types of animal cells and reported that cells has had a thin outer layer which is today known as the plasma membrane. He also concluded, based on his studies on plant tissues, that the presence of cell wall is a unique character of the plant cells. On the basis of this, Schwann had proposed the hypothesis that the bodies of animals and plants are composed of cells and product of cells. Schleiden and Schwann together formulated the theory, cell theory. This theory, however, did not explain as to how new cells were formed. Rudolf Virchow, 1855, first explained that cells divided and new cells are formed from pre-existing cells. Omnis cellula e cellula. He modified the hypothesis of Schleiden and Schwann to give the cell theory a final shape. Cell theory, as understood today, is all living organisms are composed of cells and product of cells. All cells arise from pre-existing cells. An overview of cell. You have earlier observed cells in an onion peel or human cheek cells under the microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cells of the human cheek have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane-bound structure called the nucleus. This nucleus contains the chromosomes, which in turn contain the genetic material DNA. Cells that have membrane-bound nuclei are called eukaryotic, whereas cells that lack a membrane-bound nucleus are called prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both the plant and animal cells. Various chemical reactions occur in it to keep the cell in the living state. Besides the nucleus, the eukaryotic cells have other membrane-bound distinct structures called organelles like the endoplasmic reticulum, Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. The prokaryotic cells lack such membrane-bound organelles. Ribosomes are non-membrane-bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cells, Uh, Ribosomes are found not only in the cytoplasm but also within the two organelles, chloroplast and mitochondria, on rough endoplasmic reticulum. Animals cells Animal cell contain the another non-membrane bound organelle called centrosome, which helps in cell division. Cells differ greatly in size, shape, and activities. For example, mycoplasmas, the smallest cells, are only zero point 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. The largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular. The shape of the cell may vary with the function they perform. This is a red blood cell, round and biconcave, and this is the white blood cell. It is called amoeboid. This is columnar epithelial cells, long and narrow. This is a nerve cell, branched and long. This is a trachea, elongated. Mesophyll cells, round and oval. Diagram showing different shapes of the cell. Prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma and PPLO, pleuropneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cells. They may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, rod-like, coccus, spherical, vibrio, comma-shaped, and spirillum, spiral-shaped. The organization of the prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shapes and functions. All prokaryotes have a cell wall surrounding the cell membrane except in mycoplasma. The semifluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically innate, not enveloped by a nuclear membrane. In addition to the genomic DNA, the single chromosome or circular DNA. Many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmids. The plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistant to antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryotic cells except for ribosomes. Prokaryotes have something unique in the form of inclusions. A specialized differentiated form of cell membrane called the mesosome is the characteristic of prokaryotes. They are essentially enfoldings of the cell membrane. This is a typical eukaryotic cell. Size is 10 to 20 micrometer. This is a typical bacteria, 1 to 2 micrometer. This is a PPLO, pleuropneumonia like organism, about zero point 0.1 micrometer, and viruses, zero point 0.0 to zero point 0.2 micrometer. Diagram showing comparison of eukaryotic cell with other organisms. Cell envelope and its modifications. Most prokaryotic cells, particularly the bacterial cell, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure. The outermost glycocalyx, followed by the cell wall and then the plasma membrane. Although each layer of the envelope performs distinct function, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of the differences in the cell envelopes and the manner in which they respond to staining procedure developed by Gram-Vis. Those that take up the gram stain are gram-positive, and the others that do not are called gram-negative bacteria. Glycocalyx differs in compositions and thickness among different bacteria. It could be a loose sheath called the slime layer in some, while in others it could be thick and tough, like capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membranous structure is the mesosome, which is performed by the extensions of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamella. They may help in cell wall formation, DNA replication, and distribution to daughter cells. They also help in respiration, secretion processes to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes, like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores which contain pigments. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extension from their cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hoop, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides flagella, villi and fimbriae are also surface structures of bacteria but do not play a role in motility. The villi are elongated tubular structures made of a special protein. The fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rocks in streams and also to the host tissues. Ribosome and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made of two subunits, 50S and 30S units, which when present together form 70S. Prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysome. The ribosomes of a polysome translate the mRNA into proteins. Inclusion bodies. Reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie free in the cytoplasm. Example: phosphate granules, synophysin granules, and glycogen granules. Gas vacuoles are found in blue green and purple and green photosynthetic bacteria. Eukaryotic cells. The eukaryotes include all the protist plants, animals, and fungi. In eukaryotic cells, there is an extension, compartmentalization of cytoplasm through the presence of of membrane-bound organelles. Eukaryotic cells possess an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structures. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as the former possess cell walls, plastids, and a large central vacuole which are absent in animal cells. On the other hand, animal cells have centrioles which are absent in almost all plant cells. I'll come to this diagram later by first understanding the organelles. Let us now look at individual cell organelles to understand their functions and structure. Cell membrane. The detailed description or the detailed structure of the membrane was studied only after the advent of the electron microscope in the 1950s. Meanwhile, chemical studies on the cell membrane, especially in human red blood cells (RBCs), enabled the scientists to deduce the possible structure of plasma membrane. These studies showed that the cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer sides and the hydrophobic tails towards the inner part. This ensures that the non-polar tail of saturated hydrocarbons is protected from the aqueous environment. In addition to phospholipids, membrane also contains cholesterol. Later, biochemical investigation clearly revealed that the cell membranes also possess proteins and carbohydrate. The ratio of protein and lipid varies considerably in different cell types. In human beings, the membrane of erythrocyte (RBC) has approximately 52% of protein and 40% of lipids. Depending on the case of ease of extraction, membrane proteins can be classified as integral and peripheral. Peripheral proteins lie on the surface of the membrane, while the integral proteins are partially or totally buried in the membrane. This is the fluid mosaic model of plasma membrane. This is the integral protein and this is the peripheral protein. These are the sugars, carbohydrates, and this outer layer, this is the polar head and this is the non-polar tail, which is hydrophobic, which needs protection from the outer aqueous environment, and these tiny yellow things are cholesterol in between the non-polar tails of fats or lipids. An improved model of the structure of cell membrane was proposed by Singer Sanger and Nicholson, 1972, widely accepted as fluid mosaic model. According to this, the quasi-fluid nature of lipids enables lateral movement of proteins within this overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the point of view of functions like cell growth, formation of intercellular junctions, secretion, endocytosis, cell division. One of the most important functions of the plasma membrane is the transport of molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called as passive transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient from higher concentration to the lower. Water may also move across this membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. As the polar molecules can't pass through the non-polar lipid layer bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient from lower to the higher concentration. Such a transport is an energy dependent process in which ATP is utilized and is called active transport. Um, its example is sodium or potassium pump. Cell wall. As you may recall, a non-living rigid structure called the cell wall forms an outer covering for the plasma membrane of fungi and plants. Cell wall not only gives shape to the cell and protects the cell from mechanical damage and infection, it also helps in cell-to-cell interaction and provides barrier to undesirable macromolecules. Algae have cell wall made of cellulose, galactans, mannans, and mineral like calcium carbonate, while in other plants it consists of cellulose, hemicellulose, pectins, and proteins. I repeat, algae have cell wall made of cellulose, galactose, no, galactans, mannans, and minerals like calcium carbonate. The cell wall of a young plant cell, the primary wall, is capable of growth, which generally or gradually diminishes as the cell matures, and the secondary wall is formed on the inner towards membrane side of the cell. The middle lamella is a layer mainly of calcium pectate which holds or glues the different neighboring cells together. The cell wall and the middle lamella may be traversed by plasmodesmata which connect the cytoplasm of neighboring cells. Endomembrane system. While each of the membranous organelles is distinct in terms of its structure and function, many of these are considered together as an endomembrane system because their functions are coordinated. The endomembrane system include endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. Since the functions of mitochondria, chloroplast and peroxisomes are not coordinated with the above components, these are not considered as the part of endomembrane system. Now this is an assertion reason question. Endoplasmic reticulum. Electron microscope studies of eukaryotic cells reveal the presence of a network are called SER, smooth endoplasmic reticulum. RER is frequently observed in the cells actively involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The SER is the major site for synthesis of lipid. In animal cells, lipid-like steroidal hormones are synthesized in SER. Golgi apparatus. Camillo Golgi, 1898, first observed densely stained reticular structures near the nucleus. These were later named Golgi bodies after him. They consist of many flat, disc-shaped sacs or cisternae of 0.5 to 1 micro diameter. These are stacked parallel to each other. Varied number of cisternae are present in a Golgi complex. The Golgi complex or the Golgi cisternae are concentrically arranged near the nucleus with the distinct convex axis of the forming phase and concave trans or the maturing phase. The cis and trans phases of the organelle are entirely different but interconnected. Let's see the Golgi apparatus and endoplasmic reticulum first. This is the endoplasmic reticulum. This is the nucleus, and these are the nuclear pores. This is the rough endoplasmic reticulum because of the presence of ribosomes on it, which synthesize protein, and the smooth endoplasmic reticulum because there are no ribosomes on it, which synthesize lipid in animals. This is Golgi apparatus nearer to the nucleus, and these foldings are called cisternae. The Golgi apparatus principally performs the function of packaging materials to be delivered either to the intracellular targets or secreted outside the cell. Materials to be packaged in the form of vesicles with the ER fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. And the maturing phase is concave or trans, or maturing phase is that. And what do you call cis phase? Cis phase is the forming phase. This explains why the Golgi apparatus remains in close association with the endoplasmic reticulum. A number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of Golgi apparatus before they are released from its trans phase. Golgi apparatus is the most important site of formation of glycoproteins and glycolipids. Lysosomes. These are membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes called hydrolases, lipases, proteases, and carbohydrases. Optimally active at the acidic pH, these enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. Vacuoles. The vacuole is the membrane-bound space found in the cytoplasm. It contains water, sap, excretory product, and other materials not useful for the cell. The vacuole is bound by a single membrane called tonoplast. Yeah, uh, there's chloroplast, so there's also tonoplast, which bounds vacuole. In plant cells, the vacuoles can occupy up to 90% of the volume of the cell. In plants, the tonoplast facilitates the transport of a number of ions and other materials against concentration gradients into the vacuole. Hence, their concentration is significantly higher in vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation in excretion. In many cells, as in protists, food vacuoles are formed by engulfing the food particles. Mitochondria. Unless specifically stained, are not easily visible under the microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cells. In terms of shape and size also, considerable degree of variability is observed. Typically, it is sausage shaped or cylindrical, having a diameter of 0.2 to 1 micrometer, average 0.5 micrometer, and the length 1 to 4.1 micrometer. Each mitochondrion is a double membrane bound structure with the outer membrane and the inner membrane dividing its lumen distinctively into two aqueous compartments, the outer and the inner compartment. The inner compartment is filled with a dense homogeneous substance called the matrix. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of enfoldings called the cristae towards the matrix. The cristae increase the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Mitochondria are the sites of aerobic respiration. They produce cellular energy in the form of ATP, hence, they are called powerhouses of the cell. The matrix also possesses a single circular DNA molecule, a few RNA molecules, ribosome 70S, and the components required for the synthesis of proteins. The mitochondria divide by fission. So this is the mitochondria, structure of mitochondria, a longitudinal section. This is the outer membrane and this is the inner membrane. This is the matrix, intermembrane space, and these are the cristae, enfoldings. Plastids. Plastids are found in all plant cells and in euglenoids. These are easily observed under the microscope as they are large. They bear some specific pigments, thus imparting specific colors to the plants. Based on the type of pigments, plastics can be classified into chloroplast, chromoplast, and leucoplast. The chloroplast contain chlorophyll and carotenoid pigments, which are responsible for trapping light energy essential for photosynthesis. In the chromoplast, fat soluble carotenoid pigments like carotene, xanthophylls, and others are present. This gives the part of the plant a yellow, orange, or red color. The leucoplasts are colorless plastids of varied shades and size, which store nutrients like amyloplasts store carbohydrates, starch, potato, example. Elioplast stores oils and fats, whereas aleuroplast stores proteins. And majority of the chloroplast of the green plants are found in mesophyll cells of the leaves. These are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles having variable length of 5 to 10 membranous tubules called the stroma lamella, connecting the thylakoids of a lumen of the different granule. The membrane of the thylakoids encloses a space called a lumen. The stroma of the chloroplast contains enzymes required for the synthesis of carbohydrates and proteins. It also contains small double-stranded circular DNA molecules and ribosomes. Chlorophyll pigments are present in the thylakoids. The ribosomes of the chloroplast are smaller, 70s, even in mitochondria it was 70s, than the cytoplasmic ribosomes, which is 80s. This is the sectional view of chloroplast. Outer membrane, inner membrane, granum, thylakoid, stroma lamella, and the stroma. Ribosomes. Ribosomes are the granular structures first observed under the electron microscope as dense particles by George Palade, 1953. They are composed of ribonucleic acid and proteins and are not surrounded by any membrane. The eukaryotic ribosomes are 80s, while the prokaryotic ribosomes are 70s. Each ribosome has two subunits, larger and smaller subunits. The two subunits of 80s are 60s and 40s, while that of 70s are 50s and 30s. While Swedberg's unit stands for the sedimentation coefficient. It is indirectly a measure of density and size. Both 70s and 80s are composed of two subunits. This is the ribosome. Large subunit, small subunit. Cytoskeleton. An elaborate network of filamentous proteinaceous structures consisting of microtubules, microfilaments, and intermediate filaments present in the cytoplasm is collectively referred to as the cytoskeleton. The cytoskeleton in a cell are involved in many functions such as the mechanical support, motility, maintenance of the shape of the cell. This is the cilia and flagella. First we learn about. Cilia and flagella are hair-like outgrowths of the cell membrane. Cilia are small structures which work like oars, causing the movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for the cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that of the eukaryotic flagella. The electron microscopic study of cilium or the flagellum show that they are covered with the plasma membrane. Their core, called the axonum, possess a number of microtubules running parallel to the long axis. The axonum usually has 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of axonal microtubules is referred to as 9 plus 2 array. The central tubules are connected by bridges and is also enclosed by a central sheath, which is connected to one of the tubules of spokes. The peripheral doublets are also interconnected by linkers. Thus, there are 9 radial spokes. Sorry for the little confusion. The peripheral doublets are also interconnected by linkers. Both the cilium and flagellum emerge from centrioles-like structures or the basal bodies. This is the structure of cilia or flagella showing. This is the electron micrograph and this is the diagrammatic representation of the inner structure. This is the plasma membrane. peripheral microdoublets, an interdoublet bridge, central microtubule, radial spokes, central sheath, centrosome and centrioles. Centrosome is an organelle usually containing two cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar materials. Both the centrioles and inner centrosome lie perpendicular to each other, in which each has an organization like the carpet. They are made up of 9 evenly spaced peripheral fibrils of the tubulin protein. Each of the peripheral fibril is a triplet. The adjacent triplets are also linked. The central part of the proximal region of the centriole is also proteinaceous and called the hub, which is connected with tubules of the peripheral triplets by radial spokes made of protein. The centrioles form the basal body of cilia and flagella and spindle fibers that give rise to spindle apparatus during the cell division in animal cells. Nucleus. This is the last topic. Last second topic. Nucleus as a cell organelle was first described by Robert Brown as early as 1831. Later, the material of the nucleus stained by the basic dyes was given by the chromatin named by Fleming. The interface nucleus of a cell, when it is not divided, as highly extended and elaborate nucleoprotein fibers called chromatin. Nuclear matrix and one or more spherical bodies called nucleoli. New electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 15 nanometer called the perinuclear space, forms a barrier between the materials present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears ribosomes on it. At a number of places, the nuclear envelope is interrupted by minute pores, which are formed by the fusion of its two membranes. These nuclear pores are formed by the fusion of its two membranes. These nuclear pores are passages through which movement of RNA and protein molecules takes place in both directions between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell. Variations in number of nuclei are also frequently observed. Can you recollect the names of organisms that have more than one nucleus per cell? Some mature cells even lack nucleus. Example, erythrocytes, RBCs of many mammals and seed tube cells of vascular plants. Would you consider these cells as living if they are enucleated? Absolutely not. Because they are functioning right now and they are cells. And as it already shown, if there is one cell present, that is considered as living. Here is distributed among its 46 or 23 pairs of chromosomes. We will study the details of DNA packaging and the formal of a chromosome in class 12. Yeah, every chromosome visible only in dividing cells essentially has a primary construction or the centromere on the sides of which disc-shaped structures called kinetochores are present. Centromere holds two chromatids of a chromosome. Based on the position of the centromere, the chromosome can be classified into four types. The metacentric one chromosome has the middle centromere forming two equal arms of the chromosome. Submetacentric chromosome has centromere slightly away from the middle of the chromosome, resulting in one shorter arm and one longer arm. In case of acrocentric chromosome, the centromere is situated close to its end, forming the extremely short and one very long arm, whereas the telocentric chromosome has a terminal centromere. I was talking about this types of chromosome based on the position of centromere. You can see this is the satellite and this is the secondary construction. This is the centromere as it's just moving forward and forward and forward. These two are short arms and even these two are short arms and these are quite long arms. The centromere is slightly moving towards the upper sides of the arms. Sometimes a few chromosomes have non-staining secondary constructions at a constant location. This gives the appearance of a small fragment called the satellite. This is the satellite. Last topic, microbodies. Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal cells. And there was one organelle that was not present in plant but was present in animal. What was that organelle name? That was centrioles. Now let's talk about the diagram. This is the plant cell in which rough endoplasmic reticulum. This is the rough endoplasmic reticulum because it has ribosomes. This is the lysosome that was produced by Golgi apparatus. This is the Golgi apparatus. And this is the smooth endoplasmic reticulum as it doesn't have any ribosomes on it. These are plasmodesmata. Microtubule, these are microtubules. Peroxisomes, cytoplasm. And this is the vacuole consisting of 90% of the cell. This is the middle lamella. This is the cell wall. And this is the nuclear envelope, nucleus itself, and nucleolus inside the nucleus. Plasma membrane. Remember, middle lamella is the outermost layer, but in between the plasma membrane and cell wall. This is the mitochondria. Actually, there are many mitochondria, but this is mitochondria singular. These are ribosomes, chloroplast. That's it. That concludes the plant cell. And this is the animal cell, distinct from the plant cell. This is the Golgi apparatus. This is the mitochondria. These are the ribosomes, rough endoplasmic reticulum, nucleus, nucleolus, nuclear envelope, smooth endoplasmic reticulum. microvilli, and a centriole which is absent in plants.